Clearing the malignancy. Clearing the malignancy.
In this video, I'm going to explain the consequences of zinc deficiency, as well as the consequences of calcineurin deficiency as a result of zinc deficiency, leading up to iron trapped in macrophages, which can be a problem. Okay, so basically, zinc deficiency impaired calcium influx via TRPML1 failure. So the normal state of lysosomal calcium releases mainly through TRPML1 channels, which the activity of those channels depends on proper lysosomal membrane integrity. With zinc deficiency, TRPML1 gating becomes unstable, and lysosomal calcium release drops, and therefore the cis-cytosolic calcium spikes needed for signaling do not occur. So low calcium means calcineurin remains inactive due to calcium not rising. Also, calcineurin cannot dephosphorylate NFA2 or NFAT. Therefore, no IL-2 replenishment for exhausted T cells after ongoing CD27, CD70 axis. This is where lysosomal biogenesis stops, which will lead to iron trapped in macrophages, because inactive calcineurin means TFEB locked in cytosol. So TFEB is short for master lysosomal transcription factor. If it's phosphorylated, it will stay stuck in cytosol because the inactive calcineurin did not remove the phosphate groups. So therefore, calcineurin deficiency, master lysosomal transcription factor remains phosphorylated and does not translocate to the nucleus. This results in no new lysosomes and no new renewal of lysosomes. Therefore, failure of lysosome biogenesis leading to iron trapped in macrophages, because macrophages handle iron through lysosomal recycling. Also, no lysosomes means no iron recycling enzymes. Okay, ferritinophagy requires functional lysosomes. Heme degradation, or heme is iron, requires lysosomal mitochondrial coupling. Without lysosome renewal, ferritin accumulates, iron stays sequestered as Fe3. Ferroportin export decreases. The outcome, iron locked inside macrophages. Functional iron deficiency in blood and tissues. So even with adequate dietary iron, it becomes biologically unavailable, just because of a lack of lysosome biogenesis. Inflammation can make this worse because inflammatory signaling upregulates hepcidin.
Hepcidin traps iron inside macrophages, preventing its release into the bloodstream. Vitamin D activates its receptor and blocks hepcidin production.
For reducing ferroportin. So hepcidin goes up, ferroportin goes down. That means that ferroportin cannot release or allow iron to escape from macrophages. So increased reactive oxygen species furthers TRPML1 dysfunction. So that's why it's important to have antioxidants. Also, TRPML1, being dysfunctional, cannot properly allow calcium efflux because calcium is needed for calcineurin. And if calcineurin cannot, phosphate groups from NFAT, IL-2 cannot be replenished for T cells. Also, AP1 is needed as well. So what seems to be needed for IL-2 replenishment is, for T cells, is, okay, so basically, CD70 on the tumors, they have adhesion molecules, which allows the CD70 to show on the tumor. So those have to be cleaved. Those molecules could be integrin, talin, kindlin coupling. Possibly pineapple could cleave those therefore reducing surface expression of CD70, because that surface expression was the reason that IL-2 was missing in the first place. Antioxidants to reduce reactive oxygen species is needed as well. Also reducing hepcidin, because hepcidin going up means ferroportin and iron cannot work to take the iron out of the macrophages. Okay, there are required cofactors and steps and nutrients. Okay, for lysosome, calcium release via TRPML1, zinc, magnesium, vitamin A, and omega-3s. Calcineurin activation requires calcium, magnesium, zinc, vitamin B6. Also, avoid excessive copper to avoid the copper reducing the zinc. Okay, so lysosome biogenesis function, iron controlled, not excess, hydrolase synthesis. So copper is also needed for oxidative enzymes inside lysosomes. But of course, the copper can't be too high because it will prevent iron problems or iron processing issues within the lysosomes.
Lysosomal membrane permeabilization, or LMP. When oxidative stress exceeds the lysosomal membrane peroxidation threshold, membrane integrity fails. Leakage initiates programmed cell death. Green beans, rich in polyphenols, may modulate the LMP threshold.
Also, other things needed for lysosomal biogenesis and function is sulfur amino acids, such as cysteine and methionine, and vitamin B12 and folate. So get the intrinsic factor for the vitamin B12, vitamin A for the parietal cells, so that parietal cells will secrete intrinsic factor for vitamin B12. Okay, now what's needed for iron release from macrophages is zinc for ferroportin expression, vitamin C to turn iron into more the bioavailable or more stable form, which is Fe2. So change Fe3 iron to Fe2 iron. Vitamin A for macrophage differentiation, and then glycine for heme synthesis and export. Okay, also, calcineurin deficiency leads to iron hypoxia and cancer metastasis. Also, iron becomes sequestered or misrouted into exosomes, leading to stabilization of HIF-1 alpha, therefore hypoxia, even when oxygen is present. So cancer cell thrives on hypoxia. Another major issue with a lack of a lysosome biogenesis is that the MVB that are loaded with oxysterols will not have a lysosomes to fuse to, because if MVBs can do so, and if the MVBs don't have lysosomes to fuse to, the MVBs will end up fusing to the plasma membrane, allowing exosome release, and then the exosomes will be free to wreak their havoc and damage on the immune system, even at the chromatin DNA level. So when zinc is deficient, calcineurin is suppressed, therefore reduced number in lysosomes. Also, no tethering fusion machinery, which is HOPS and SNARE, HOPS and SNARE, because that machinery is needed for fusion of the MVBs to the lysosomes. But since the machinery will be missing due to calcineurin deficiency, the cells will resolve this problem by fusing MVBs with the plasma membrane, which is not good because that will release the exosomes. Okay, so due to the TRPML1 channel not functioning or not shuttling the calcium correctly, the calcineurin is deficient and therefore no lysosomes. So quick recap, zinc deficiency means less TRPML1 for calcium, therefore less calcineurin activation, persistent phosphorylation of NFAT because calcineurin did not remove the phosphate groups, therefore NFAT excluded from the nucleus, but the NFT is supposed to translocate into the nucleus. Therefore, a reduced lysosomal biogenesis, resulting in iron trapped in macrophages because lysosomes are not present or functional to deal with iron. Also, less lysosomes for fusing with the MVB. Therefore, the MVB, which are sacs inside of other sacs, they end up releasing exosomes, which wreak a lot of havoc because the MVB had to fuse to the plasma membrane because there were no lysosomes to fuse to. So that's it for the video. Like, comment, share, subscribe, and I'll see you in the next video.